Meiosis turns diploid into haploid. So this is where the number of chromosomes is halved. So imagine n is the number of chromosomes, okay? This is simple algebra here. 2n turning into n means it's divided by 2, it's halved. This is why meiosis is a reduction division, because it halves the number of chromosomes, or n, okay? And now, before meiosis and mitosis, DNA replication must happen before. I have a video for this as well. Let's start with meiosis, with meiosis 1, prophase 1. There's also a meiosis 2, which we'll look at as well. First thing that's going to happen, chromatin condenses into chromosomes, okay? And the nuclear membrane disintegrates, as you're going to see here on the left. Boom, nuclear membrane disintegrates. Now, these chromosomes are the following. They're going to be the joining of two sister chromatids, sister for identical, so they have the same alleles, they have the same genes. So these are going to form what's called a bivalent. So this is a bivalent, bi for two, okay, held together by a centromere. It's this circle in the middle, this black circle is a centromere. This whole thing is a bivalent of two sister chromatids joined together. And now, before anything else, crossing over is going to happen. As you can see here, these sister chromatids are identical because they're, well, sister chromatids. Red one and red one, and blue one and blue one. They are identical. But they will no longer be identical because between different chromosomes, there's going to be an exchange called crossing over. And this exchange involves the exchange of DNA segments, DNA fragments, okay? So these swap, and because of that, well, first they swap by the chiasma, and because of that, the sister chromatids are no longer genetically identical to one another, and they become what's called recombinant chromatids. Now, the chromatids that are not involved in crossing over and are still genetically identical are what we call non-recombinant chromatids. So make sure you note that down as well. And finally, we can see that gametes, so the sperm cell and the egg cell, inherit different combinations of alleles from the maternal, from the mother, and paternal, sorry, chromosomes, and this process is thanks to crossing over during prophase 1 of meiosis. Back in prophase 1, we can see that centrosomes, or microtubule organizing centers, produce bundles of microtubules called spindle fibers. So these centrosomes, or MTCOs, produce these spindle fibers right here, this purple thing. And centrosomes are for animal cells, and plants involve microtubule organizing centers, or MTCOs, and these both have the same function, which is producing the bundles of microtubules called spindle fibers. And have in mind that centrosomes contain something called centrioles. At the end of prophase 1, this is what the cell is going to look like, more or less, okay? So the sister chromatids, well, they are no longer sister chromatids. The recombinant chromatids are going to be around the center, okay, of the spindle fibers. The centrosomes or MTCOs are positioned to the sides. And now we move on to the metaphase 1, the second phase of meiosis 1. Okay, here the spindle fibers are going to attach to the centromeres. So the purple thing is going to attach to the black circles of homologous chromosomes. So now we call them homologous chromosomes. Okay, so as you can see, they attach to the purple thing. So they're lined up by the equator of the cell. You know, Earth, there's the equators. It's a similar shape. Anyways, the maternal and paternal homologs show random orientation towards the poles. This is very important. This links to natural selection. about how um, genetic diversity is formed. So this also happens in the metaphase 2 of meiosis 2, further increasing genetic diversity. And this is called independent assortment, where the positions of maternal to paternal and maternal to maternal chromosomes are independent of one another. Random orientation. Keywords linked to natural selection. Now, anaphase 1, after all this random orientation, independent assortment, and lining up by the equator of the cell, we have that the following is going to happen, okay? These spindle fibers have shortened, as you've just seen, and they have separated the bivalent, or tetrad. Now, these are going to pull homologous chromosomes apart and towards opposite poles. Okay, so these two have been pulled to the right pole, these two have been pulled to the left pole, and this is the difference. Moving on to telophase 1, the cell has elongated, as you just seen, and the homologous chromosomes reach the poles and decondensed. Okay, I couldn't represent how they decondensed, couldn't find an animation, but essentially they've reached the poles, the ends of the cell after it has elongated, and the spindle fibers have pulled them here. Now, these spindle fibers are actually going to break down into phase 1, as you just seen, and a nuclear membrane that disintegrated in prophase 1 is going to form again around each nucleus of the new two cells we're going to have the division. But before that, there's interkinesis, which is the period of rest between meiosis 1 and meiosis 2. Moving on to meiosis 2 now, after cytokinesis. Cytokinesis 1, it's simply the division of these. Okay, so before the cells used to have 4 of these homologous chromosomes, and now the cells have 2. So the number of chromosomes halved, and this is why meiosis is a reduction division. As always, any questions can go in the comments. Prophase 2. Okay, so we're starting with meiosis 2. What's going to happen? The nuclear membrane disintegrates in both cells. The homologous chromosomes recondense. Again, I couldn't show an animation for this. I'm sorry about that. Just remember that. And MTCOs migrate to opposite poles, as you've just seen right now. Spindle fibers start to form. Okay, not fully. They fully form by metaphase 2. Perfect. Now these spindle fibers attach to the centrosomes, as you've seen here, the centrosomes of these homologous chromosomes, and they show random orientation towards the pole. The poles, remember, this is independent assortment. Again, this further increases genetic diversity. Anaphase 2, what happens? Oh, the spindle fibers have shortened, as you see in these purple things have stretched to the sides. They shortened. They split the centromere, so you can see the black circle isn't here anymore. It's split these in two, and it's pulled the sister chromatids, well, not necessarily sister chromatids, the recombinant chromosomes, apart towards the opposite poles of the cells, the opposite ends. And now once these are separated, they're called chromosomes. Oh shit. Anyways, telophase 2. Again, the cells elongate. Well, now it's cells. Before it used to be cell in telophase 1. The chromosomes reach the opposite poles of the cells and decondensed, and the nuclear membrane forms around each nuclei. What happens next? You guessed it. Cytokinesis 2. Well, you don't necessarily need to call it cytokinesis 1 or cytokinesis 2. It would just be cytokinesis for both cases. So it's going to happen. Boom. 4 cells. We went from diploid to haploid. And again, the number of chromosomes has divided. It went from 8 chromosomes to 4 chromosomes, and now 2 chromosomes per cell. Wow. 4 haploid daughter cells is the final product of this division. We know that cytokinesis happens in post-meiosis. It also happens post-mitosis, as we're going to see later. But in animal cells, it's going to be the following process, okay? So there's going to be an equal cytoplasmic division, which means that the cytoplasm and its contents are going to be divided equally in both daughter cells, which are the same size, okay? When the parent cell splits and makes these 2 daughter cells, this is going to be the case, okay? Ensuring equal structure and function of both of them. Now, both daughter cells will receive the following. At least 1 mitochondria for aerobic respiration or the production of ATP, okay? Oxygen goes in, CO2 goes out, and then there's also glucose and water involved, okay? Now, any organelle only creatable through division, such as peroxisomes, okay? So some organelles cannot be created when the cell is already born, okay? So the cell cannot produce them. They can only be produced through division. So the daughter cells will obviously need to receive these if they want to live on. Now the animal cell is a network of actin and myosin proteins which form a contractile ring, okay? Or it contains a network of actin and myosin proteins, okay? Now this contractile ring pinches the cell membrane together and it forms what we call a cleavage furrow. Now this is facilitated by the double membrane. Let me actually underline that if it's possible. Yes, sir. So what happens here? How do animal cells split? Well, this is the microfilament ring, which, thanks to the contractile ring, forms the cleavage furrow, this right here, where they're starting to separate, but they're not fully separated. And then it turns into the division of the parent cell. This would be the parent cell, the original cell, which has elongated, and then the cleavage furrow has formed, and it forms the 2 daughter cells, which have an equal cytoplasmic distribution. Now, enucleation links to this, okay? What is enucleation? Well, it's the lack of a nucleus, not having a nucleus. Now, before cytokinesis, nuclear division is needed, okay? Why? Because it prevents producing anucleate cells, or cells that do not have a nucleus. Now, we know that red blood cells don't have a nucleus, so they can fit in more hemoglobin and carry more oxygen, fine. These naturally anucleate cells, such as red blood cells, lose their nucleus after differentiation by enucleation. So we know that all cells in our body have the same DNA, but they are different because some genes are activated and some are not. That's gene expression, okay? And that causes the differentiation. Now, red blood cells actually initially have a nucleus. It's just that it eventually gets removed to fit the space for the hemoglobin, and this is the process of enucleation. Note that down. Moving on to cytokinesis, but this time in plant cells, okay? We're still looking at eukaryotic cells because plants, animals are both eukaryotes. Differences here is There's no cleavage furrow, no contractile ring. There's a cell plate, which is formed by the fusion of vesicles containing cell wall material. Okay, forms like a mini cell wall in the middle of the cell plate. Okay, here it's starting to form. Eventually, it forms, and then the daughter nuclei are established. There's a new cell wall, and notice how they're not separated. They're like still stuck together. Well, this is how leaves are formed, right? Like they're still stuck together, and um, they are different cells, but uh, they're not going to be like fully separated, unlike in animal cells. Oogenesis is the process of producing mature egg cells or ova in human females. This involves the primary oocyte or the oocyte 1, which splits into the first polar body, and the secondary oocyte or the oocyte 2. Okay? And note down that both of these have an unequal distribution of cytoplasmic contents. It's unfair. Why? Because the secondary oocyte has all the cytoplasmic content, while the first polar body has basically none. Okay. Now, this secondary oocyte, via fertilization, turns into the second polar body and the mature ovum. Okay. And now, the mature ovum contains all the cytoplasmic content that it needs for embryonic development, because we know that this mature ovum uh, eventually is fertilized when the sperm cell uh, enters it, and it forms the zygote. And we know that the zygote turns into the embryo, I think, and that turns into X, that turns into Y, and eventually you get the fetus and the baby forms. Okay, so this is how you were born, ladies and gentlemen. Uh, Now, the unequal distribution of the cytoplasmic content that we see from the oocyte 1 to the oocyte 2, and then the first polar body, provides the necessary nutrients for embryonic development and ensures proper chromosome count, which should be haploid, N, okay? We know diploid is 2N, so it needs to be haploid. Budding is a form of asexual reproduction in fungi, okay? And it starts with the parent cell doing the following. Ooh, what happened? It forms a developing bud, which turns into a new bud, which turns into a budding scar, this hole right there. And here you have a new cell. I'm pretty sure the animations before lagged, but I think you see what I mean. Like a bud formed from the parent cell, and then it split, and then it formed the budding scar. Wow. Finally, it's time for mitosis. There's going to be a prophase, as always, first phase, where chromatin condenses into chromosomes and the nuclear membrane disintegrates. But watch out, this is not the same as in meiosis, okay? There's little details. Like, for example, that crossing over doesn't happen in prophase 1, and so then there's no genetic diversity. The daughter cells are genetically identical. So chromatin condenses into chromosomes. The two sister chromatids join together to form a bivalent, held together by a centromere. Okay, and again, crossing over does not happen here. Okay, moving on to the metaphase. What happens here? Well, spindle fibers attached to the centromeres of homologous chromosomes, as you can see here. These spindle fibers should be attached to this black circle here. My bad. Not the best thing, but anyways, anaphase. Oh, what happened? What just happened? Well, these spindle fibers have shortened, as you've just seen, and this pulls these sister chromatids apart. Okay, we know that the spindle fibers used to be stretched out to the middle, and boom, they go closer to the poles, pulling these sister chromatids with them. Now in the telophase, the cell is going to elongate in preparation for cytokinesis. Okay, now the spindle fibers are going to disintegrate, as you might have seen. Okay, yeah, the spindle fibers disintegrate, and the chromosomes are going to decondense. Crazy. They're also going to move to opposite poles, and the nuclear membrane is going to reform at each of these poles, and then eventually cytokinesis is going to happen again. Now, you need to be able to identify the different phases under a microscope. So, prophase 1, right here, where the nuclear membrane is disintegrating. That's why it has this texture. Then metaphase 1, where they line up, okay? The spindle fibers have formed here, and they're holding at the sister chromatids by their centromeres. The anaphase, where they split the sister chromatids apart, so the spindle fibers shorten, okay, stretch out, bring the sister chromatids to opposite poles, and the telophase that we've just seen, where the cell is starting to elongate and the nuclear membrane is starting to reform, okay, and chromosomes have also decondensed. So all this diagram explanation I've given here, it can be applied to what you see in these microscope diagrams. Now that we've looked at both meiosis and mitosis, we can outline the similarities and differences. Starting with the differences, here are the boxes on top. Meiosis is a reduction division because it goes from diploid cells with 2n number of chromosomes to haploid, to 4 haploid cells with n number of chromosomes. So the number of chromosomes has been halved, as I said before. These are genetically unique, okay? And that's what we want for our gametes, the sperm and the egg cells that we need for sexual reproduction. Why are they unique? Well, this is thanks to crossing over from prophase 1 and independent assortment from metaphases 1 and 2. Okay. Now, mitosis occurs in somatic cells, not in germ cells, which are gametes, okay, in somatic cells. Somatic cells are basically any type of cell which is not a sperm or the egg cell, which is not a gamete. The word for that are germ cells. It produces diploid 2n daughter cells, which are genetically identical to the parent cell, because here crossing over and independent assortment, independent random orientation do not occur. This produces cells for asexual reproduction. Okay, we know that bacteria reproduce asexually and they produce genetically identical offspring unless mutations occur. Cells for growth. So when you're growing up in puberty, your cells are uh, well reproducing via mitosis, okay, and replacing lost or damaged cells. Now, what are the similarities? So, in both of these processes, microtubules will be involved, which are the proteins forming the spindle fibers or the spindle apparatus, okay? Now, these spindle apparatuses, the spindle fibers, pull the chromosomes apart, okay, as we can see from the anaphase 1, the anaphase 2, and the anaphase of mitosis. DNA replication must occur before both. They are both nuclear divisions, though the only reduction division is meiosis, okay? Uh, cytokinesis, sorry, will happen after both of them. Okay, to separate into different cells. And finally, microtubule motors are present in both. These are specialized proteins that bind to the microtubules or the spindle fibers and they hydrolyze ATP. Okay, so basically they use ATP because hydrolyzing is turning ATP into ADP to move the chromosomes to either pole or the ends of the cell. And that would be the similarities and differences. Let's look at some harmful mutations. Okay, first one, trisomy, which is the inheriting of one extra chromosome. Try for 3. So one extra, if you usually have 2, well, if you have one extra, that would be 3. So having 3 instead of 2 chromosomes inherited. Okay. Type of trisomy, trisomy 21, also known as Down syndrome, which is having an extra copy of chromosome 21. Now, non-disjunction is an issue involving the failure of pairs of homologous chromosomes to separate or disjoin, for disjunction in anaphase 1 of meiosis. And this can lead to monosomy X or Turner's syndrome, which is a gamete being produced without chromosome X in meiosis. We know the meiosis produces gametes or germ cells, sperm and exo. So if these are missing chromosome X, this is called Turner's syndrome or monosomy X. And we can see that through karyotyping and a karyogram, which is a separate topic. You need to know that DNA in eukaryotic cells, so animals, plants, fungi, is in the nucleus, okay? Where nucleosomes, this thing right here, are going to contain the DNA. How? Well, first note that nucleosomes are the building block of chromatin. This links to mitosis and meiosis. Note that down. But going back, how does it contain the DNA? Well, the DNA, as you can see, is supercoiled around 8 histone proteins. So the histone proteins are these blue balls you see here. You can see there's 8, and the DNA is this orange or red strand. Okay, it's the double helix. Now you can see 8 here, but there's actually a 9th one. Okay, there's a 9th histone protein which stabilizes the DNA. Now, how are these actually held together? Well, this is because DNA has a negative charge. This is going to link with gel electrophoresis that we'll look at later. So remember, DNA, very important, has a negative charge, while the histone proteins have a positive charge. And this leads to, if you do chemistry, electrostatic attraction, which is basically the attraction of negative to positive. And now, note that in prokaryotic cells, such as bacteria, the DNA is what we call naked in the cell. Now you know that DNA is supercoiled around the histones through electrostatic attraction. But what does this actually benefit? Well, it's going to ensure that the DNA is stuck somewhere, right? But this makes it more efficient to separate during nuclear division, which is meiosis or mitosis, right? They're both nuclear division. Meiosis is the only reduction division here. Now, this is only during nuclear division because when the cell is not in cell division, the DNA is actually loosely packed around the histone proteins. It's not like super coiled, very hard, okay? And again, during nuclear division, the chromatin, so remember the nucleosomes make up the chromatin, is supercoiled and condensed by the histone proteins. And this turns it into a chromosome, okay? We can see chromatid here and chromosome here. This would be what it looks like.